Dear friends, today we are going to discuss one important and interesting topic, maybe a debatable topic, that is truth in a pill. During our lifetime, we need to take pills, medicines. In this 20 or 30 minutes, we'll try to dissect some informations about a specific aspect of pill. And for that, this presentation may contain some controversial issues. I need your valuable comments, your valuable feedback on those issues in the comment box or chat box. So let's start today's journey with truth in a pill. So when we are talking about truth, it is important to acknowledge this great lady. And this is a quiz. I will pause here. I request you to think the name of this great lady. One clue for you: by the virtue of this great lady, due to her concern regarding truth in some specific pill, she had saved thousands of mothers to face this type of consequences, which had affected Europe and world a lot. Now I know you guess who is she. She is Dr. Frances Kathleen Oldham Kelsey. So Dr. Kelsey was a determined FDA scientist who had saved American children from this catastrophe, that is thalidomide disaster. But her journey starts long back. You can see this picture in your left side. Dr. Gelling and Dr. Kelsey, this was in the 1938, just after doing her PhD in pharmacology. He had joined FDA as scientist, and she was investigating the elixir of sulfanilamide related atrocities. And due to her enormous effort and research, with the help of Gelling and other scientists, the US or world, I think, gets something called as a drug law, which had regulated the development of the drugs in US. So US Congress passed the Federal Food Drug Cosmetic Act of 1938. Now she had done his also medicine career in 1950s, and after that she had joined FDA in 1960 in a very high rank position, and her first assignment was to review the application from the Recursion and Merrill Company for approval of the thalidomide, which was used in Europe that time in a lots of pregnant mother to control the nausea or morning sickness. And we all know the consequence of using this molecule there in Europe. The total number of people affected by the use of this thalidomide during pregnancy was estimated at more than 10,000. And unfortunately, 40% died at or shortly after the time of birth. And 60% of them had an unfortunate event. They had phocomelia, no limbs, or absent limb, partial limb. So this is why till date, Dr. Kelsey was worshipped there in US. She had got the highest rank of US award too. And 1962, the Kefauver-Harris Amendment happened, and US law, US drug regulation become more stringent. So I think when we are talking about truth in pill, before starting the journey with truth in a pill, we should salute this great lady who had saved a lot of Americans from this bizarre consequence from thalidomide. Now, very recently, this book was published. First of all, the disclaimer is like that. I am not going to say you to read this book, or I am not going to say, please do not read this book. I am from a neutral point of view. I am just telling you this was the book I found when I was preparing this topic. And this is something interesting. And one very important quote that I found, that bitter truths about Indian drug regulatory architecture, which calls for some soul searching by policymakers. So we need to know about the truth, because truth is a bitter pill to swallow also. So maybe I'm not going to discuss what is written in this book, but we will try to make our presentation on the issues like this, how the drug development happen compared to generic development, what is actually the development process of innovator. Then we'll also try to discuss about how we can call a generic is therapeutic equivalent of innovator. Of course, as I had mentioned, there will be lots of controversies, lots of challenges with this generic. We'll talk about some case studies and also a few considerations with some specific innovator brand we'll try to discuss. I'm again saying there could be a point of view, opponent point of view, and I'm welcoming all those opponent point of view, because it is also important to respect the otherness of others. So first of all, when we are talking about pills, it is important to know about the nomenclature of drugs. So a same drug, there could be a multiple names, just like our Sri Krishna. How many names he had? So if we consider the different name, it could be the drug name, that means the international non-proprietary name. So that is totally non-proprietary. For an example, it could be pioglitazone, it could be atorvastatin, etc. It could be amlodipine. Now, when one innovator company is bringing some specific drug, so they will have another name that will be given by the innovator. For example, pioglitazone, the Takeda company brings it, so it was Actos. The Pfizer brings Lipitor, which is atorvastatin, like this. And also we have a IUPAC name, the International Union of Pure and Applied Chemistry. So that is some chemical name is there. I am afraid I cannot remember those type of names. I am not a chemistry professional now. Another group of nomenclature is brand names. So here, for the brand names given by the generic manufacturers, we can call it as branded generics. So they are generics, but the name is given by the generic manufacturers. So that is the brand names. Here we can see for pioglitazone, it could be a Pioglar or Pios by USP, like this. So as we are talking about drug development and we are talking about new drug development, it is very important to acknowledge it is a complex and costly procedure, taking almost 10 to 15 years. And there are multiple studies conducted that suggest the cost between 800 millions to 2 billion US dollar to get a new drug to market. And this is how an innovator drug come into the market. And when we are talking about innovator drug, the journey starts with some new chemical entities, which is almost 5,000 to 10,000. And after that, when we get in a molecule which is ready for clinical trial entering into human, so from 5,000, it drops down to 5 to 10 new chemical entities. And after this investigational new drug submission happened to FDA, FDA will give permission for clinical trial. So there will be phase 1, phase 2, and phase 3 clinical trial. Then they will submit here for new drug application to drug regulators. There will be the regulatory review. They will check all the data of different phases of clinical trials, and then they will approve the drug. Post-approval, there will be another trial. So that is a phase 4 clinical trial. So when we talk about the generic drug development, they do no need to follow this all process because they already know the chemistry. The basic active pharmaceutical ingredients will be the same. So they only need to submit here, not the new drug application like this NDA. They have to submit here the abbreviated new drug application. And for that, they do not require this whole lot of things. So it is important to understand we need generics because obviously the cost factor and also the accessibility. So when we term affordability, the affordable healthcare, that should consider both the accessibility and the availability. So accessibility and the affordability both are important. So also we need to control the market monopoly to reduce the health-related burden of the country, and also we want to export to other countries. But what is actually the generic medicines? Ideally, by definition, they should contain the same active ingredient as the originator molecule. Identical in strength, dosage form, and the route of administration with the same use of indications. They should be bioequivalent. They should meet the batch requirements for identity, strength, purity, quality, like that. And they should follow the strict standards of US FDA's or some specific drug regulators. For example, in India, CDSCO's specific good manufacturing practice regulations required for the original products. But at the same time, the inactive ingredients may vary. So this was what we were discussing for a new drug, for an innovator drug to come into the market, they require detailed animal studies, clinical trials, and of course, during the clinical trials, they need to do some bioavailability studies. But here, the abbreviated new drug application for generics, they require only the bioequivalence studies. So to approve generic, the state drug regulators should check whether the generic manufacturers are following the good manufacturing practice guidelines or not. After that, they need to submit this in vitro dissolution study. What is that? How the release of active pharmaceutical ingredient on certain dissolution or liberation characteristic is happening, and their comparison with the innovator that had been examined. And finally, the in vivo studies of bioavailability and bioequivalence, specifically noting pharmacokinetic parameters like Cmax, Tmax, or area under the curve checked during this generic approval process. But now the question is, when we are considering a complex of cardio-renal metabolic disease in diabetes, and we have one innovator here, this Foxiga, which has a specific API we all know, the propanediol monohydrate salt of dapagliflozin, which is a crystalline format. But when we are talking about other dapagliflozin, a lot of dapagliflozin is now available in India. We do not know the active pharmaceutical ingredient. We know about the dapagliflozin is there, but with whom? And how is their formulation? Whether it is crystalline every time or amorphous. Now, when we are talking about Foxiga, when we are talking about dapagliflozin, that is a crystalline formulated formulation is there, they had been gone through all these robust landmark clinical trials, including declared to DAPA-HF or DAPA-CKD, and had shown some specific results which had compelled us to write this molecule for those complex cardio-renal metabolic disease patient. So whether just having one report of maybe a BAB study which may contain around 15, 16 person can give us the confidence of writing it. So that is the question to you. Of course, generic medicines are cheaper. They are typically 30 to 80% cheaper than the originator equivalents. But the question, the universal question is whether the quality and performance of generic drug is comparable to brand drugs. So that is the question. When we talk about equivalence of generics, this is based on some regulatory assumptions. And what is that? If we have a pharmaceutical equivalence proven and bioequivalence proven, altogether we can say this drug is therapeutic equivalence to innovator. So this is based on regulatory assumptions. So when we talk about pharmaceutical equivalent, it is important to know that they should contain the same active ingredient, same dosage form, same route of administration, identical in strength or concentration, but they may have some differences too, which is acceptable by the regulators, like shape, the release mechanism, some extent labeling, scoring, and most importantly, the excipients. Also the color, flavors, and preservatives. So that is something important. And when we talk about generics, this unfortunate story always comes into our mind. And this was a quote from a famous uh, paper which had uh, quoted like this: to suffer from CML is bad enough, but to suffer for being taking some substandard medicine is totally unacceptable. So we know that how these adulterated drugs had uh, caused some unwanted uh, penalties, or we can say that the India's scenario, India's dream to become the pharmacy capital of the world was first time being uh, hammered by this story. And we want to forget this, but of course we need to learn from this too. But at the same time, in 2019, there was another paper which had suggested with the same type of molecule, imatinib. So that paper suggests this particular original research had suggested the change from original to generic imatinib appears to maintain the efficacy and be generally safe. More patients and longer follow-up are required to confirm these observations. But this paper gives us some ray of hope too with generic drugs. So at the same time, if we are talking about generic drugs, it is important to consider the quality of the manufacturing. If we maintain a good quality, good pharmaceutical equivalent, then I think that would be helpful for our patient. So similarly, another systematic review on similar type of molecules, imatinib, the efficacy and safety in CML. So in this particular systematic review, the authors had suggested generics can be comparable to the branded imatinib. They have a similar efficacy and safety profiles, but manufacturer variability may impact the findings. So of course, generics reduce cost and increase the accessibility, but at the same time, we need to be cautious on this variability, the manufacturer's variability. Now coming to this in vitro dissolution to in vivo BAB testing, some new brand which compares the release of active pharmaceutical ingredients on this dissolution liberation characteristics like in vitro dissolution studies and also in vivo BAB studies to check whether the Cmax, Tmax and area under curve are matching with the reference standard or not. But in the dissolution study, when we are talking, this classification system, biopharmaceutical classification system, is important to note. We have 4 classes of drugs. So if we have a very high permeability and solubility, so that is class 1. But if the solubility is low but permeability is high, then that is class 2. Class 3, they have low permeability but high solubility. And class 4, they have both the low permeability and low solubility. So according to US FDA, all class 1 drug who have the high permeability and high solubility, they can have the bio-waiver. So bioequivalent study need not to be done in those class 1 drug. But in India, the guidelines are a little bit broadened. So even the class 1 and class 3 drugs. The class 3 drugs mean who have low permeability but high solubility, they also can be waive the bioequivalence study. So that can be a clause to have the bio waiver. So that is something arising questions, because the molecules like dapagliflozin, rosuvastatin, specifically the dapagliflozin, because that is a BCS class 3 drugs. So some generic dapagliflozin can get into our market, can get the regulator's permission without having the proper BABE study also. So they will not do the BABE study and they will come into the market. So that is something we need to know, specifically when we are prescribing drugs like this dapagliflozin or drugs like even the empagliflozin. Some very recently the linagliptin generics will be also coming. So they all are BCS class 3 drugs. Already we have sitagliptin. So there will be a possibility that many companies can bring this drug without doing the proper BABE study. So that is something we need to know. Now, as we are talking about BABE, let's know about little bit about this concept of bioequivalence. So if the rate and extent of absorption of the test drug do not show a significant difference from the rate and extent of absorption of the reference drug when administered at the same molar dose of the therapeutic ingredient under similar experimental conditions in either a single dose or multiple dose. So I am applying the same drug and I am checking whether the rate and extent of absorption, how they are entering into our systemic circulation, whether it is comparable to the innovator molecule. That is the main purpose of bioequivalence study. And if we consider the guideline for our BABE study, so what is there? If the confidence interval of those area under curve Cmax, that values should not be rounded off. Therefore, to pass a confidence interval range between 80 to 125. And if the value should cross that 80 line, that means it is below 80%, then it will not be a bioequivalent. Or if it is more than 125%, then it will also cannot be a bioequivalent. So my take here is, you can see in this diagram, in this cartoon, this is the innovator molecule, branded innovator molecules, you say. And now this is one generic X and this is another generic Y, where both of them can be approved because this is between this 125%. I think this is the upper confidence interval is below 125%, and the lower confidence interval is more than 80%. This is also the same. But whether they are all equal by regulators, by the criteria of bioequivalence study, yes, they are equal. So this is something also can cause some itching in my brain, whether it is actual. When we are talking about Cmax, Tmax, and area under curve, we need to understand this is the maximum plasma drug concentration, which is called as Cmax. So even if a 10% difference of Cmax, and that can cause some impact, specifically when we are taking some low dose molecules, like for an example, levothyroxine. some microgram dose. So even we are having a 10% drop or 20% above the dose, that can cause something huge differences in this Cmax. Maybe the Tmax, the time required to achieve a maximal concentration, or area under curve is the total area under the plasma drug concentration time curve. So this is what we need to be a little bit cautious. And for that, the other agencies like EMA, US FDA, or even the Japan and Canada, they are now developing another important criteria, and that is narrow therapeutic index drugs. So normally it is 80 to 125% for them, for all the other aspects, but they are giving one specific criteria as narrow therapeutic index drugs, that is between 90 to 111. So the confidence interval is very narrow here. But the problem is, in India, there is no narrow therapeutic index drug. So 95% confidence interval of the ratios of like area under curve, Cmax, Tmax, that should fall between this 80% and 125% from the branded drugs. Here you can see the branded drug here. Now, if the upper confidence interval, the lower confidence interval is below the 80% line, so that will not be a non-equivalent, that will not be an equivalent, bioequivalent. Even if it is the upper confidence interval is more than 125%, then also that will not be a bioequivalent, it will be a high non-equivalent. So for bioequivalent, that should come under this scenario, between 80 to 125%. But here, particularly in this diagram, what is important to note, if assuming this is for Cmax, so the Cmax of this bioequivalent generic and Cmax of this branded drugs, they are bioequivalent, and they also, if we just check their Cmax, they do not have any statistically significant difference. But in case of this scenario is also possible according to the bioequivalence guideline. So here you can see the generic 2 and generic 3, they are also bioequivalent, but they are having statistically significant difference with the reference product. Whereas G5 and G6, they are also generics, but are they all equal? This G2, G3, G5, G6 are equal? Can you say like that? So that is the question. Yes, according to the regulators, they all can be called as generics because they comes under this 80 to 125%. But that is a huge range. So almost 45, the difference is 45%, 80 to 125. So whether it is actually acceptable, specifically when we are treating some disease which do not require any form of variability in effect. So that is something we need to explore. There are certain criteria for narrow therapeutic index drugs. These drugs should be tested under that criteria. But the question is, and that for that chart, this this is from the US FDA side. But we do not have this type of site, this type of narrow therapeutic index drug. So that is also an appeal to our regulators to please make some guidelines or regulations for those drugs which have narrow therapeutic index. For developing generics, they also should follow the international guidelines, and the confidence interval range should be between 90 to 111. So that is a request to our regulators. Though the anti-diabetic agents, pharmacologically, they cannot be defined as narrow therapeutic index drugs. But as we always being taught about diabetes management should also consider how to reduce the glycemic variability. So whether when we are considering our anti-diabetic agents, it is important to have a stringent criteria like 90 to 111 for developing generic for anti-diabetic agents, not a broad confidence interval range like 80 to 125%. So that is also a thought I want to share with you all to think about it. So we know about why the generic and brand name L-thyroxine are not interchangeable. And specifically, if we want to interchange it, we should be cross-check with the TSH value. Ideally, the guideline says we should not change the brand. So brand substitution should not be done. Even a small differences between LT4 formulations can cause significant change in TSH level. And that is what we were discussing. That if I am having a very small drug, then a small change in the Cmax, maybe 10%, that can cause a huge effect. So that is something we want to consider. Now, this is just a question to you all, that a student A got 100 marks, a student B getting 80 marks, and student C is getting 64 marks. Now, someone say that A, B, C, all of them should get the first prize. They're equal. Is it okay with you? So in Indian B study guideline, what we have, we told about that 80 to 125%, but with whom we can compare? So it is not always. We need to choose the innovator drug as a reference product. So this is the latest update, 2020. Almost now it is 4 years. We don't know when we will get the updated list. But this list suggests if you have atorvastatin generics to be marketed in India, then you can compare it with the innovator brand along with some other branded generics too. So there is also no specific guidelines. We do not know how they are selecting these reference drugs. For an example, as we were talking about that 100, 80, and 64. So the innovator, they are the comparator for the first time. So if we consider it as 100 marks, that their Cmax is 100 something. Now we got one generic, that is B, and that generic have a 80% of Cmax of the drug A, so that is 80. And after maybe some years, we don't know how they are selecting because there is no clear-cut guideline laid in any documents available. So if that same B is now becoming the reference product, and we are bringing another drug, that is C, as generic, and what happened? We are taking B as reference product, and it could be the C which may have a 80% of the Cmax of B and get into the market because they are fulfilling that 80 to 125% criteria. So ultimately, if we compare the Cmax of C to Innovator, that is A, so that the C is 64% of the Innovator. So Innovator, if it is 100, the Cmax of C will be 64. So it could be a possibility. We don't know because we do not have any specific guidelines on that. So also, it should be our demand to our regulators. We should be very transparent about this selection criteria of reference product. Otherwise, the prescribers can have the confusion. Why they will write the generic? Yes, in India, we need generic medicine. There is no doubt in that. But as a prescriber, it is also our right to know the right message. And for that, we need to know the truth. So this is also under demand towards our regulators. And we don't know whether this is the cause why there are some Indian studies where we found the generics are working not like the innovator drugs. So this is one example on latanoprost. And another very recently, this study was published on itraconazole, generic in itraconazole vis-a-vis innovator, where there is less action and also the therapeutic drug levels were higher in innovator compared to the generic itraconazole. So generic itraconazole, they have less effect, and that less effect is due to that they are not reaching the therapeutic drug levels as the innovator. So this is one case published in long back in Journal of Psychiatry and Neuroscience, where a 74-year-old man diagnosed with hypertension on amlodipine. The BP was not reduced that much. So family physician give them the pindolol, and that BP was controlled. And after 2 years, again, the blood pressure went above the later figures, and he had some fainting spell in the apartment. BP was 220 and 180 again. So what happened? The medication compliance was good, but there were a switching to generics, and they were suspecting that switching was causing this rise in BP. So this type of issues happened not only in India, but also in Western countries. So another important issue is like that when we are talking about the active pharmaceutical ingredient in a pill, there is an important part which is inactive, that is the excipients. But excipients also can cause side effects. A very common example is the cross-armellose. This cross-armellose sodium is used as an excipient in furosemide. And there were some cases where the innovator furosemide was not doing the allergic reaction, but the generic furosemide was causing. And the cause was due to this cross-armellose. And this was an event we all know, the outbreak of this anticonvulsant intoxication in an Australian city. What was there? The 87% of them, the blood phenytoin levels were above the therapeutic range. When They had searched, they found the phenytoin peel, when they use lactose as excipient, then the level of blood phenytoin level is going high. But when it was calcium sulfate, it is going down. So here you can see how the influence of lactose is there. Whereas in the calcium sulfate excipient, the phenytoin level is going drop. Again, with lactose, it going high. So it is important to consider that excipients may not always be biologically inert, and that is why we need to be very cautious while using, or rather while switching from innovator to generics. We need to know about the excipients too. And this was again a very sad event which had occurred very recently. The Gambia and Uzbekistan killed lots of babies because of this adulteration. And that is the diethylene glycol and ethylene glycol, which we all know can cause acute kidney injury. And this is another stroke against our reputation as pharmacy of the world. So we need to be very cautious. So let's have some data with dapagliflozin, which is a very renowned anti-diabetic agents we all know now. But very recently, there is a loss of patent there, and we are getting multiple generics. We got generics. They are proven bioequivalence, and sometimes some generics, as already discussed, may enter into the market without doing the BAB study because of that class 3 DCS characteristic of this dapagliflozin. But what is important to understand, for any organic molecule, the spatial arrangement, that is very critical. They are different than another. If one atom is shifted in any three-dimensional direction by even 1 degree, and because if just we shift them in this 3D direction, that can open up a very broad possibility of different other molecules, each with their unique placement of atoms in three-dimensional space. And that is the concept of chirality. That is the mirror image, non-superimposable molecules. And to say that a molecule is chiral is to say that its mirror image, it must have one, is not the same as itself. We know lots of examples are there, like levosalbutamol, like S-amlodipine, etc. So the important thing is that the enantiomers of a chiral drug may differ significantly in their bioavailability, rate of metabolism, metabolites, excretion. So the pharmacokinetic, pharmacodynamic characteristics can be different. We do not have detailed data on the dapagliflozin generics, but what is important to note, this is also a chiral molecule, and it has 5 stereogenic centers. So having the right stereogenic center is the key for appropriate pharmacokinetic, pharmacodynamic property of a molecule. We need to acknowledge this, and that is why sometimes we need more data beyond BAB study for molecules like this, which had shown multiple good effects in different systems. We know about its action on glycemia. We know about its action on heart, kidney. But whether that can be simply transcribed into all the generic dapagliflozin, where we do not have any data on its spatial rearrangement, the enantiomer characteristic, that is another question where I want to end. And when we talk about the statins data, that is something interesting, that when we talk about atorvastatin, generics that obtained from multiple sources worldwide that contain a methylated impurity and which reduces their HMG-CoA reductase inhibitory effects. So this is a concern with statin generics, the methylated impurity, and that changes from brand to brand. So a real-world evidence study done with statins, and this was a huge data, large data, almost 14,000 records were included and analyzed. And interestingly, it was found when compared to the innovator statins, the adjusted probability of all-cause death was significantly higher with generics, 62% with previous cardiovascular events. So that is something need to be concerned. And when we talk about stability of any drug, we need to know there are certain impurities like lactone, anti-isomers, 5-keto acids. So these impurities can be formed during or after production of the rosuvastatin. And these also can impact the stability of the rosuvastatin formulation and overall the shelf life. So when prescribing rosuvastatin, how many of us consider this typical pharmaceutical characteristics of this particular pill? So that is also something we need to consider, because that may impact the efficacy and safety. We have the Spain data, but yes, we do not have any Indian data here. And maybe it is important to give stress on generating Indian data, whether our generics are as good as our innovator molecules. That if we can get from some, that information if we can get from some hard evidences, real world evidences, so that can be beneficial for us. So of course science matters, there is no alternative to innovation, but it is also important that experience also matters, and innovator is, that is why is not always equal to generics. So we need to consider that too, the experiences, the evidences, like big cardiovascular outcome trials, big heart failure trials, big renal trials, CKD trials. But at the same time, as professional, we all should support to help our future too. We need to plan how we can have a data, how we can have we can generate some information on generics, that yes, that generic they also are equal clinical. According to the regulators, they are equivalent therapeutically. There is no doubt in that. So not everyone can afford this. Also, we need to consider, I am writing one drug and the patient is not buying because of cost, and that leads to some event, bad event. So that is also not desirable. So overall, decreasing our healthcare expenditure is important, but at the same time, we need to improve the standardization. So in India, we know our former president had mentioned this, India's pluralism is the greatest strength. But at the same time, in India's pluralism, the comprehensive thinking with a focus on standardization is also essential. It is not like that we cannot use different type of products, different types of concentration. There should be a uniformity. So be safe and choose wisely when you are prescribing a pill. So that, I think, the message I want to give you. And if you have any difference in opinion, you can write in the chat box and we can take those opinion too. So thank you. Thank you once again for hearing me.